Hello, everyone. Welcome to our first lecture of the genetics course. This is the first class of seven or six other ones that are coming next. Um, because this is our very first class, I wanted to give you some general idea of what to expect for these recordings. Okay, so first of all, you will see that at the bottom of my slide, there's probably some closed captions. Okay, I do not guarantee that they are going to be 100% correct because I do have an accent, and sometimes the program that I use doesn't fully understand what I'm saying. But I do try to use them regularly to make these classes or these recordings more accessible to those that need them, right? And sometimes it's nice to see um, the captions as well. I do like to watch videos with captions. I think it helps me with the understanding of the concept. Also, another point about these recordings or these slides is that all the pictures that you will see, they have a alternative text, meaning that if you use a software that reads those images, you will be able to listen to a alternative text to them. If you do have an issue with the accessibility of these lectures, I do ask you to come talk to me. I want to make sure that your learning experience is maximum, that you do learn the concepts that we are going to be working throughout these classes. So if you do have any issues that may require some additional accommodation or any other resources, please feel free to come talk to me. Okay? In general, the way I try to organize these recordings is by welcoming you to this class and going through my slides. Sometimes you will see that I will take notes on those slides, and I do recommend that you do the same, okay? Because only listening or just looking at my slides might not be enough for some of you. It's not enough for me. I know that for sure that I need to actively take notes of what the person is talking. Otherwise, I will completely forget everything. So please do take notes as I go through the slides because you will find that very helpful when you are studying. Okay, especially because I might, ask you something in the exam that I mentioned in class or that I pointed out when I was explaining the content. So create the habit of taking notes, right? This very first class you will find as a not as exciting, not as very genetics because it's a first class, and I want to make sure that all of us are on the same level of understanding about the cells, about the DNA, because that's going to be the foundation for you to understand the next concepts in this course. So I decided to make the first class about mitosis, meiosis, and also about the DNA, proteins, and all that stuff that you've probably seen in your introductory biology courses, because I feel like it's important that we see things at different levels, especially if you decide that you want to be, a doctor in the future, right? A biologist in the future. And if you're following this um, health track, I think it is important for you to be very assertive and firm about these concepts. Okay, so our first class will give you a general overview about the DNA, the macromolecules, uh, and cell cycle. All right, but I do want to start by giving you a perspective of how the history of the of the genetics and cellular biology evolved throughout uh, time. And I want to start by saying that science became interested about, cells, about the development of the organisms, and about how traits are passed throughout generations because people were interested about or understanding about how life is created, right? We had in the past many explanations for how life was generated. And the understanding about life and how life is generated has changed throughout the history of science. But that, served as a fuel for many of the advancements that we have in the area of genetics. Okay, so we can certainly agree that genetics, started very early in the history of science because even though the concepts such as genes, chromosomes, DNA, RNA, and all those fancy names, they only show up in the 19s, right? In the 19th, in the in the 19s. The interest about the interest for understanding those molecular bases of life were very early in the history of science. Okay, so at the very beginning of these studies, and of course, I'm not giving you the whole story because there's a whole bunch of things. I'm just gonna highlight some of the main findings and some of the main theories. But the main idea that people had in the past was that um, a human being was formed from was originated from a fertilized egg that contained a complete, already formed miniature adult. So that fertilized egg was called homunculus. And you probably have seen or heard about this theory before, which is called the theory of perform, performationism. I'm sorry about the English. You will see me struggling throughout the classes with some of those fancy words. But this theory of perfashion, perf, per, perf, preformationism, I'm sorry, preformationism would propose that we already existed, but in a miniature version inside of a fertilized eggs, of a fertilized egg. All right? That's what people believed, but that doesn't necessarily reflect the truth of how development occurs. It is only with William Harvey and his theory about epigenesis that he will say that no, actually, a adult is not preformed in a miniature inside of a fertilized egg, but this adult comes from successive developmental stages from that fertilized egg. So that fertilized egg will go through developmental stages and become a adult organism. And that explanation was also extrapolate was also um, used to explain not only for humans development, but also other living beings development. Okay, and of course you can see how those two theories, the preformationism and the epigenesis theory, they were very conflicting with each other because one would say, okay, no, the human is already preformed inside of a fertilized egg, and the other one would say, no, this human comes from successive developmental events, developmental stages, right? And his theory by William Harvey is even further supported by the cell theory that only came in the 1800s. And these two guys, they actually discovered that organisms, that living organisms, they are composed of cells. And those cells come from pre-existing cells. So one thing added to the other in this theory of epigenesis. The knowledge about the existence of cells and the knowledge about these um, developmental stages, okay, that this living being has to go through to become an adult organism, okay? This is the picture of what people knew as homunculus. All right. So you have a fertilized cell, and a fertilized cell would carry a miniature human inside. And that homunculus, right, would develop um, into a more adult version. Okay. As if the baby was already inside of that fertilized egg. Okay. Another theory that existed to explain how living beings are created was the theory of spontaneous generation. This theory was created when when sanitation was a problem in the past. So people didn't actually cared about cleaning their clothes or cleaning their homes or anything like that. So it was very common in Europe, leaving piles of clothes. Um, and people would observe that after a few days, rats, cockroaches, mice would come out of that pile of clothes. So they thought that those living beings were spontaneously generated by dirt, right? So a non-living component gave rise to a living creature. But that wasn't the case. We understand now that those rats, those cockroaches were there because what? Because it was dirty and they liked to be in those dirty clothes, right? But people would believe that non-living things would give rise to living organisms. Not until Louis Pasteur came with his idea and with his work about the existence of microorganisms and microbes. So Louis Pasteur was a very interesting scientist because he refused to believe that things were spontaneously generated, right? So he developed a series of experiments, and there's a ton of information about him on the internet, and I do recommend you to uh, go and watch those things and read those things because it's very interesting. He developed many experiments, and he never proved, or he wasn't able to prove that non-living things would give rise to living organisms. But he found out that for life to exist, you need a pre-existing form of life. That makes sense, right? You need a pre-existing living creature to give rise to a newly existing creature, right? So he came with this idea that, you know, this living organism needs something before to exist. And it's just fascinating how his work, contributed with understanding about vaccines, microbes, and microorganisms, and understanding even about washing your hands, for instance. It's just fabulous. I do recommend you to look into his story a little bit more. His idea with, associated with the epigenesis theory is also then further added with the work by Mendel and the work developed by Darwin. Those two guys, they add significant contribution to our understanding of how living beings are created and how traits in those living beings are passed through generations. Okay? And their theory set the stage for our current understanding about development, about evolution, and about genetics. So Mendel and Darwin would be very important. Their work would be very important because they start providing more explanation for those phenomena that people were um, observing in the past. Darwin, let's start with Darwin. Darwin was the guy who formulated the theory of natural selection. So Darwin observed that the existing species, they came from ancestral species. That idea that a living being comes from another pre-existing living being. Okay? But he observed that this new living being came with modifications compared to the ancestral species. Just like you came from your mom and dad, but you don't look like them. I mean, sorry, you look very similar, but not identical to them, right? Because you're modified somehow. So he observed that the new offspring had some differences compared to the pre-existing species. And those modifications were actually a response to natural pressures in the environment. So the environment would pose pressure to those subjects, to that species, to those individuals, and that would select the fittest organism of that environment. And that organism would then leave descendants, would then create a offspring with a little bit of modification. Let's think of a very dumb example now that comes to my mind. Of course, this is not trying to reduce the work by Darwin, okay? So let's say that we have in this environment a um, butterfly with orange wings, a very fluorescent, orange wings. Very easy prey, right? Very easy prey. So that butterfly won't actually leave descendants because, bigger animals will eat those butterflies. So they can't pass their traits, that orange fluorescent color, to the offspring. But a mechanism, and we are going to discuss about those mechanisms, a mutation happens that some of these butterflies start to have black wings. And through generation, those that have darker wings, they survived. First of all, for that butterfly to exist, it needed a ancestral species, right? Before that. Second, that new individual, that new butterfly is not the same as the pre-existing butterfly. So is a descendant with modification. And that modification occurred because the butterfly was suffering a pressure from this environment. Okay? That altogether helped him to formulate this theory of natural selection. The theory of natural selection is an explanation, provides an explanation for how evolution occurs. So evolution, think of evolution as a big umbrella. Okay, evolution is a biological phenomenon that happens in nature. Darwin formulated an explanation for how evolution occurs, and that would occur through natural selection. Okay, it's important that you start understanding that natural selection is not the same as evolution. Okay, natural selection seeks to explain how evolutionary changes occur through generation. Okay, this is very important because it adds to our, or it added in the past, our understanding about, how traits are passed, not how traits are passed, I'm sorry, about how those traits are modified, right, through generations, and how you needed a pre-existing living being to generate the current existing living being. Okay, but only Darwin, only Mendel will actually offer a more quantifiable explanation for how those traits are passed along through generations. So if Darwin studied these natural selection mechanisms, Mendel provides a explanation for how, a quantifiable explanation for how those modifications are passed through generations. Okay, again, I am reducing a lot of the history. I'm trying to tell you the history of centuries in about what, 30 minutes or so of class. So please keep in your head that things were more complicated than just what I am um, trying to explain here. Okay, so Mendel will be very important because he provides, he's the one that sets the stage for the development of this genetic field in biology. Okay, to understand how those traits pass throughout generations can be quantified, and how we can study those traits and how they influence the phenotype observed in the subject. Okay, so Mendel, with his experiments, he provides a quantitative data that shows how those traits are passed from the parental to the offspring. Okay, and we now can predict how those modifications can occur. So Mendel and his work will form the foundation of what we have now for genetics. Okay, we know now that these Mendelian factors, and this is going to make more sense in the other classes, but these factors, these traits, are in the genes, and the genes are in the chromosomes. We now know that, but Mendel didn't know anything about the existence of chromosomes when he developed his work. He didn't know where those traits, where those features were in the cell. Okay? It's only later on that other scientists will formulate, will come up with this chromosome theory of inheritance. These guys, they were able to formulate that these traits observed by Mendel and these traits observed by Darwin and so many other people that came before them were actually controlled by the genes that were present in the chromosomes. And they also understood that these genes are transmitted through the gametes. So the gametes are the cells that contain the genetic information that is passed throughout the generations. Okay? Again, Mendel and Darwin did not know about the existence of chromosomes in the cells. It's only later on that we added their two ideas to this chromosome theory and said, okay, everything that they observed is happening at a molecular level in the chromosomes in the cells. Okay? And here, my next slide provides you a picture of these chromosomes in humans. So this is a karyotype. Okay? Karyo means nucleus, means the nuclear envelope, okay, that surrounds the chromosomes. This is a picture of how those chromosomes look like. In humans, we have 46 chromosomes, and those chromosomes are divided in 23 pairs. So here you have the first pair, second pair, third pair, fourth, fifth, and so on. Each pair of chromosome is composed of two chromosomes. Well, it's a pair, so it has to be two, right? So each pair has two chromosomes. These two chromosomes are called homologous. So this and these are the homologous chromosomes. Each chromosome now is formed by two sister chromatids. Okay, so you have 46 chromosomes. You have 23 pairs. Each pair has two homologous chromosomes. And each chromosome, when in its duplicated state or replicated state, has two sister chromatids. Okay? Those concepts are going to become more familiar to you as we go through this class. All right? I know that I've told you lots of things now, so just digest the information as we go through. Okay? Like I said, Mendel, Darwin, they didn't know about the existence of chromosomes or even DNA. It's only in the 1900s that we have a understanding of how the chromosome looks like, of how the DNA looks like, and understanding how those things look like expanded the field of genetics, molecular biology, and so on. At that point in history and today, we know that all living organisms they have the genetic material. And that genetic material is made of the nucleic acid. And the nucleic acid is the DNA. Okay? That DNA contains the genes. The genes are how those traits are written in letters. Okay? Very simple explanation of what a gene is. How those traits are written, are codified in our genetic material. All right? That DNA is organized into chromosomes, which are those structures I showed you before. The cells in eukaryotes undergo through two cellular divisions, and they are mitosis and meiosis. Mitosis happens when diploid cells originate two diploid cells with the same number of chromosomes. Meiosis only happens with the gametes or spores if we're talking about plants. Here, a diploid cell originates cells with half of the number of chromosomes, and we call those cells haploid cells. Okay, the processes, and then we are gonna look at them, are similar, but the final result is different. Okay, so mitosis creates two cells, and each of the cell has the same number of chromosome, meaning that, let me get the pen. If you have one cell with 46 chromosomes, mitosis generates two new cells, and each cell will have 46 chromosomes. However, meiosis will generate cells with half of that number. So one cell with 46 chromosomes undergoes two divisions. We have meiosis 1 and meiosis 2. We're going to talk about that. And each cell at the end, oops, sorry about that. I guess it's not working. Will have 23 chromosomes. Okay? These cells we call diploid. Oops, that was ugly. These cells we call diploid. Okay? And these cells we're going to call haploid. All right? What we can see here at the bottom picture are these chromosomes. So these chromosomes here are condensed, and we can see them as visible condensed structures. Okay? They will be replicated and then divided. All right? Mitosis generates two diploid cells. Meiosis generates four haploid cells. We're going to look at those two processes individually. There are two types of organisms. We have those called prokaryotic and eukaryotic. The prokaryotic organisms are those that do not have a nucleus. The eukaryotic organisms are those that do have a nucleus. So the genetic material of the prokaryotic organisms is dispersed in the cytoplasm, whereas the genetic material of the eukaryotic organism is inside of an organelle called nucleus. Okay, who are those prokaryotic? The bacteria, for instance, and the archaea, which is also a special type of bacteria. All right, and these eukaryotics are the protists, the plants, the fungi, and the animals like us. My next slide is showing you a very general view of a cell. And you probably know from the top of your head every single organelle shown in this picture because I am sure you've seen this before. But I just want to call your attention here to the presence of the nucleus, right? So the nucleus is the organelle that contains the genetic material. And the genetic material is the DNA, right? Here in the nucleus, we have the chromatin. The chromatin is when the chromosomes are not condensed, okay? But it's just the DNA dispersed, spread through the nucleus. I just wanted to bring you this picture because we are going to talk about mitosis and meiosis. And I think it is important for you to visualize where the process is occur, okay, and is occurring in the nucleus. This is showing you where the genetic material is located. But our understanding about the composition, the chemical composition of this generic material is fairly recent and happened in the 1900s, okay, with work, for instance, from Rosalind, Watson Crick, and so many other important researchers. All right. First of all, some scientists came to the conclusion that the chromosomes are actually made up of DNA, not protein. And it is the DNA that carries the genetic information. Okay? Later on, with work from Watson Crick, we actually were able to understand what are the building blocks of the DNA. The DNA is a macromolecule, and like any other macromolecule, the DNA is composed of smaller subunits. What are the macromolecules that you probably have studied already? Carbohydrates, fats, and proteins. All those macromolecules are made up of smaller subunits, right? Proteins, for instance, the smaller subunit are the amino acids. For the DNA, which is a macromolecule, the DNA is composed of nucleotides. The nucleotides, they are composed of a nitrogenous base, a sugar ring, and a phosphate molecule. Okay? Nucleotide is the small subunit of the DNA composed of three other things. The DNA described by Watson and Crick is actually a long ladder like molecule. So think of two strands that are complementary to each other and they twist around a axis. Okay? So let's let's think about it. So you have one strand in one side and a second strand in the other side. And they twist, you can see here that there is an X in between, right? They twist around this center, forming this double helix structure. I'm sure you already know about this, so I just wanted to remember. All right. So the nucleotides are formed by the nitrogenous bases. And we have the following nitrogenous bases. We have those nitrogenous bases that have a, let me get the laser point, that have a pyrimidine or pyrimidine ring, okay? And we have those nitrogenous bases that have a purine ring. So pyrimidine or pyrimidine ring, we have the cytosine, uracil, and thymine. For purine rings, we have guanine and adenine. Okay? Uracil is the nitrogenous base only present in the RNA. So uracil is present only in the RNA. All the other ones are present in the DNA. Okay? DNA does not have uracil. I also want to point out that the nucleotides are composed of these sugars, these pentose sugars that we call them desoxyribose and ribose. What is the difference between the desoxyribose and the ribose? The desoxyribose does not have a molecule of oxygen attached to the second carbon of its ring, whereas the ribose does have a oxygen connected to the second carbon of the sugar ring. Okay? The desoxyribose is present in the DNA, and the ribose is present in the RNA. Okay? This is called a um, pentose because we have five, and let me clean this because it's getting polluted. Because we have five carbons. How do we count those carbons? We have, first of all, the oxygen. To the right of the oxygen, we have the first carbon. Then we have the second, third, fourth, and the one sticking out is the fifth carbon. Why do I need to know the positions of those carbons? That is important because that determines how those nucleotides interact with each other, which influences in the process of the replication. Okay? So the third carbon is where you have the oxygen and the hydrogen molecules connected. And the fifth carbon is where you're going to have a phosphate group connected. So, fifth carbon, think about phosphate. Third carbon, think about oxygen and hydrogen connected. Okay? Just a reminder. So, DNA has the adenine, cytosine, thymine, and guanine, whereas the RNA has adenine, cytosine, uracil, and guanine. Okay, that's probably very familiar to you. This other slide is showing you the chemical structure um, of that DNA. So here on the left, you can see that one molecule of DNA is composed of this double helix of these two strands that are twisted around a centered axis. Okay? When I expand and identify the chemical composition of that molecule, this is what I see on the right. So, in the middle of this molecule, I have the nitrogenous bases. Each nitrogenous basis is complementary to another basis. Okay? So A is complementary to T, and C is complementary to G. In the RNA, A complements with U, with uracil. Okay? Don't forget that T is replaced in the RNA. These two bases, this pair, they are connected with each other via hydrogen bonds. So the hydrogen bonds keep those two uh, complementary base pairs connected. All right? Forming the backbones on the side of the molecule, we have the sugar and the phosphate group. Remember, the sugar has five carbons. On the fifth carbon, you have the phosphate group. And on the third carbon, you have the oxygen. You can see that this side is in the orientation 5. Let me get the pen. Is in the orientation 5 to 3. And the other side is in the 3 to 5. That is because the two strands are complementary to each other. Okay? So one goes in the 5 to 3, the other one goes into 3, from 3 to 5. All right? If I give you a picture, hypothetically, in the exam and ask you to identify what is the fifth carbon and the third carbon, you've got to identify which one has the phosphate and which one has the hydroxyl, okay? The oxygen and the hydrogen groups. To make things a little bit easier, I have another picture here showing you the chemical structure. Okay, let's look at the picture on the left first and leave the one to the right later. Again, you have in the middle the nitrogenous bases. We have thymine, guanine, cytosine, and adenine for DNA. Then you have the backbone. We call this a sugar phosphate backbone because you have a molecule of sugar and a molecule of phosphate. Remember that you have 5 carbons, and on the fifth carbon, you have the phosphate. And then on the third, you have the hydroxyl group. See that the um, phosphate of the fifth carbon connects to the hydroxyl group of the third carbon. And in this connection, the molecule loses water, okay? Because you have a hydrogen here that is lost. Remember, the three and terminates with oxygen and hydrogen, all right? But for the phosphate group to connect to that molecule, to the following molecule, you need that you need that hydrogen to leave, and then the phosphate binds. Okay, so carbon, the fifth carbon has the phosphate group. The third carbon is where the phosphate group binds and forms a molecule of water. Okay, is where those two bases bind with each other. On the right figure, I like this because besides of telling you the very same thing I just explained to you on the right, on the left, it also tells you what are the chemical interactions existent in the DNA molecule. Let's remember some chemistry here now. So, connecting the molecule of sugar and the phosphate and the nitrogenous base, we have a covalent bond. Okay? The covalent bond is linking, is binding together two nucleotides of the same strand and also the sugar with the nitrogenous base. Okay? That is important because covalent bonds are really strong, are the strongest chemical bonds that exist, right? Okay, connecting two nitrogenous bases, we have hydrogen bonds. You can see that each of the, sorry, I'm pointing to the nitrogen here. I can't see very much from my loading from my PowerPoint. Each of these nitrogenous bases, they have hydrogen here. And those hydrogen, they interact with the complementary base pair and they form those hydrogen bonds that keep those nitrogenous bases uh, together from two different strains. All right. We also have another type of interaction here called the van der Waals interactions. The van der Waals interactions, they happen in between two base pairs that are stacked on top of each other. So you have this base pair and you have this base pair, and they are kept together stacked because of these van der Waals interactions. Okay, so again, you have covalent bonds that keep the phosphate sugar connected and the sugar and the nitrogenous bases connected. You have the hydrogen bonds that keep two bases together, and you have the van der Waals interactions that keep those bases stacked on top of each other. All right? This DNA undergoes through a process called replication, meaning that one DNA creates two other DNA. This is what I am saying. One molecule of DNA will create two new molecules of DNA. Okay? This replication is semi-conservative because those two new molecules, they have half of their sequence coming from the old strand. Okay? So when we replicate here and we add a new strand, so we have a new strand here and a new strand right here, you can see that the new two molecules, so you have one molecule here, one molecule here, still has an old template of the old strand DNA. So it's a semi-conservative replication, okay? Because it conserves half of the original DNA. Does that make sense? Of the original strand. I am sure you have seen this replication process before, but I thought I would bring this back again because it is important that you understand the molecular basis of the replication process before we understand, mitosis, meiosis, and the Mendelian laws. All right. So my next slide is showing you exactly this. It's showing you how the replication process occurs. Let's try to keep this very simple, and then we add complexity to this process. Okay. So here you have your parental DNA strand. Remember, one side goes from 5 to 3, the other side goes 3 to 5 because they are complementary to each other. Okay? Here, to open this molecule of DNA, because you need to make two new molecules, we have an enzyme called helicase. The helicase breaks the hydrogen bonds in between those nitrogenous bases and opens the molecule of DNA. Okay, the opening region that you see here is now called the replication fork. Okay, here the parental DNA gives rise to two strand templates. One is known as the leading strand template, and the other one is the lagging strand template. The enzyme that manufactures a new complementary strand to this template is the DNA polymerase 3. The DNA polymerase 3 polymerizes, creates a new complementary strand to the template strand. Okay? Why do I have a leading strand and a lagging strand? Here is the case. The DNA polymerase 3 likes to build a new DNA in the sequence, in the following sequence, from 5 to 3. So when it uses this um, leading strand template, it can continuously add new nucleotides because that leading template is in the direction that the DNA polymerase likes. So the DNA polymerase 3 adds new nucleotides from the 5 to 3 ends. So the leading strand has to be in the 3 to 5 orientation, okay? So replication is happening in this direction. Oops, I thought that I was a pen, that I had a pen. So it's happening from here to here, okay? So the new strand is being formed in the 5 to 3, oops, to 3 direction. Because it's being formed in the 5 to 3 direction, the template has to be 3 to 5. Okay? Then the DNA polymerase 3 will read the leading strand continuously, all right? And follow the replication fork. However, you see that I have this lagging strand. The lagging strand is in the 5 to 3 orientation. How does the DNA polymerase fix that? Because that can't be the case, right? Because a complementary DNA sequence here for my lagging would be 3 to 5, right? But the DNA polymerase 3 does not actually like that because it likes to build a new sequence from 5 to 3. What happens is that the DNA polymerase now manufacture the DNA in chunks. And those chunks are called the Okazaki fragments. The Okazaki fragments are chunks of that DNA that the DNA polymerase is producing, is manufacturing, okay, in a non continuous way. So, whereas in the leading strand, you have the DNA polymerase reading this DNA in a continuous way, here the DNA polymerase reads the strand in a lagging manner. So it reads a little bit, then goes back, reads a little bit more, goes back, reads a little bit more, and goes back. And that is because the orientation of this strand is not complementary to what the DNA polymerase wants. Okay? The DNA polymerase creates a new DNA in the 5 to 3 sequence. So it has to use a complementary template that is in the 3 to 5. So the one that is in the 5 to 3 does not work. To fix that problem, the DNA polymerase has to follow an opposite direction of um, that replication, and in doing so, creates these chunks of DNA. Okay, Let's add more complexity to this. There is other molecules and enzymes participating of this replication process. And they are, first of all, at the beginning or at the replication fork, you have these single strand binding proteins. These single strand binding proteins, they bind to the DNA and they prevent the formation of the hydrogen bonds once the helicase cut open the DNA strand. So DNA goes first, opens the replication fork. Then you have these binding proteins that prevent the hydrogen bonds from being formed again, allowing for the DNA to stay open. Okay? We also have this primase. The primase is an enzyme that that places RNA primes onto the DNA template. Why is that the case? Because the DNA polymerase 3 has to be guided. The DNA polymerase 3 does not know where to start the replication. It only finds the start of the replication, the origin of the replication, when it finds this primer, this RNA primer. The RNA primer is actually a sequence of oligonucleotide that is placed onto the template strand by the primase. And the primase is the enzyme that puts those uh, primers. Okay. We also have the DNA polymerase 1. The DNA polymerase 1 removes the primers, okay, and replace those primers with new DNA nucleotides. And then in the lagging strand side of the replication, we have the DNA ligase. The DNA ligase, like the name suggests, will bind to nucleotides. You see that in between these fragments, you have a space that has to be connected, right? Because, again, the DNA polymerase 3 produced chunks of that replication. So the ligase binds those chunks all together. Okay? Let's take a better look at this lagging and leading strand. Remember, the leading strand is coming from 3 to 5, and the DNA polymerase creates a new strand from 5 to 3. Here is a picture of that. So the leading strand, which is this one, is going from 3 to 5, which is great because now the DNA polymerase 3 can synthesize a new complementary DNA strand in the 5 to 3 direction. However, the lagging DNA strand is in the 5 to 3 direction. And look, the DNA polymerase does not like that because the new DNA strand that the DNA polymerase produces has to be in the 5 to 3 orientation. How is the problem solved? The DNA loops around, which kind of inverts the sequence of the lagging strand template. And you can see now that the lagging strand template that once was from 5 to 3 is now 3 to 5, which is good because now the DNA polymerase 3 can create a complementary strand that is now in 5 to 3 orientation. All right? Because of that, you see that the DNA polymerase will synthesize a chunk of DNA and then has to move a little bit and synthesize another chunk, and then has to move a little bit and synthesize another chunk. So the reading of that lagging strand template is not continuously like it is for the leading strand template. Okay? If this is not clear, I just have one more thing to add to this. And this is going to be very, very, oops, sorry about that. This is going to be very, very quick if I know how to use my computer. So, this is what I am saying. You have two strands, right? They are coming from the same DNA. So, one strand is in the 5 to 3 orientation. The other one is in the 3 to 5 because that's how they complement each other. The DNA polymerase only synthesize a new sequence in the 5 to 3 orientation. So here, I'm going to get a new color. So here, what is the complement of the first one at the top? Here, the complementary strand is in the 5 to 3 direction. So that's okay. So the DNA polymerase, which is right here, we read this strand continuously and fabricates and synthesizes, the new strand in the 5 to 3 orientation. However, look at the one below. See that there is a problem now, right? Because you can't form a 5 to 3 here. Because if you do that, they are not complementary to each other, right? And that's not what we like. So what needs to happen here? Let's see if I can make this like my last picture. So what needs to happen here? So the DNA goes through a loop. So now look what happens here. With this loop, see that the DNA polymerase is going towards my left. Okay? So it's going like this. Let me get a color. So it's going like this. Okay? Because of this loop, look to what happens now. So my sequence now is, let me get some color. My sequence now is in the 3, oops, to 5 orientation. So now I can create a new strand in the 5 to 3 orientation. Is that clear? Right? So when we loop, we now reverse the position of that strand. Okay? And that DNA polymerase, we read this chunk and create a new strand. Then it moves a little bit more, create a new chunk, and reads that space and that portion, then moves and creates another chunk. Those chunks are then bound together by the DNA ligase. Okay? I understand that, and these chunks are called the Okazaki fragments. I understand that this might be a little bit complex, but I do recommend that you go over and over through this process until it is very clear in your head what we are doing here. Okay, so this is the process of replication. It is important that you understand these processes of replication because they this would be the foundation for understanding how mitosis and meiosis they occur, and then to also understand the Mendelian laws. Okay, just before we keep going, full disclosure here, you might have noticed that I am sweating a lot in this video, and that's because I am recording this from Brazil. I'm actually right now spending my vacation with my family, and it's really, really warm here right now. So, and we don't have air conditioning here, so I'm struggling with the heat. So, if you see me cleaning my sweat or something, don't bother. I literally had to take a break because I was sweating and it's so warm here now. For those who not know, it's the opposite of the US or the North Hemisphere, right? The South Hemisphere now is summer, whereas in the North Hemisphere is winter, fall going to the winter, right? So that's just it. All right, but anyways, as long as you understand the process of replication, I don't I don't see myself asking you a lot of questions about, these Okazaki fragments and explaining, asking you to explain to me why is that important. But I do want you to understand, the the fifth carbon, the third carbon, how they determine how to determine the orientation of the replication. So please make sure you review this on your own and make the drawings so you can visualize what we are talking about. Okay, so replication is one of the processes that happen with the DNA, right? But um, that DNA also goes through some other processes, and we are gonna study how those processes are. Before we go through those processes, and you probably can associate them to the central dogma of biology, I just want to briefly review the RNA, which is chemically similar to the DNA. The only difference is the sugar, which is not a deoxyribose. It's a ribose because it has an oxygen, and one of the nitrogenous bases is the uracil instead of the thymine. Okay, but everything else, all those covalent bonds, hydrogen bonds that I mentioned, they are pretty much the same. Okay, one of the things here is that RNAs are usually single stranded. So instead of having two strands and forming a double helix macromolecule, RNAs are usually single stranded. All right. So, like I said, the first process that happens with this DNA is the replication. So, the replication is when the DNA makes another DNA, and we call that replication. Okay, that DNA now serves as a template for the cell to make a molecule of RNA. And the molecule of RNA here is going to be called mRNA, that stands for messenger RNA. Okay, the process of transforming this molecule of DNA into a molecule of RNA is called transcription. Okay, transcription is when the DNA is converted into RNA. That process happens inside of the nucleus. So replication happens in the nucleus, and transcription also happens in the nucleus. All right. The product after transcription is called a transcript. Okay. So a transcript is an RNA that was transcribed from a DNA template strand. This mRNA has what we know as code as codons. Codons are three letters, are triplets, three letters that function as a code for a amino acid, right? We're going to see them soon. Then this RNA is converted into a protein through a translation process. This whole thing you probably know as the central dogma of biology, right? That describes how the DNA is converted into mRNA, which is a process known as transcription, and then how the mRNA is converted to protein, and that process is known as translation. Transcription happens in the nucleus. Replication or duplication also happens in the nucleus. Translation happens in the cytoplasm of the cell. That is the case for eukaryotic cells or eukaryotic organisms, because in the case of prokaryotic organisms, those processes are going to happen in the cytoplasm because they don't have a nucleus. So now let's look at the transcription process. Okay, so this transcription process again happens in the nucleus. In that process, the DNA is used to synthesize a RNA sequence. And that RNA sequence is usually or has to be complementary to that DNA template strand. Okay, so that process usually starts usually starts at a non-sequence of letters no, called or known as Tata box. This sequence is a promoter sequence, okay? And is a sequence where you have a repetition of the um, nitrogenous bases T and A in the DNA. Because you have this repetition of T's and A's in the DNA, we call that sequence, that promoter sequence, as Tata box. Okay, that has about 25 um, nucleotides, and then that marks the beginning of the transcription. Okay, or that is the part that's part of the promoter, which is a little bit above the starting point of the transcription. All right, as you can see here, you have the Tata box sequence, and then you have the start point for the transcription. So this whole region we call promoter. All right, this Tata box is recognized by several transcription factors. And those transcription factors, as they suggest, they regulate the expression of the genes. The transcription factors are important because they signal they signal for the RNA polymerase 2, which is the enzyme that promotes the transcription, that promotes the conversion of the DNA into a RNA molecule. Okay, so here in this figure, you can see the steps. First of all, you have the promoter sequence, and that sequence has a Tata box sequence, which is about 25 nucleotides above the starting point. This promoter serves as a signal for these transcription factors and for the RNA polymerase 2 to find where the DNA has to, where the translation, where the transcription, I'm sorry, where the transcription has to begin. All right. The process of transcription is carried by the RNA polymerase II, which then recognizes the presence of these transcription factors and then starts reading the strand of DNA. Okay? This process is less complicated. There's not a lot of enzymes involved in this process, like helicase or DNA ligase or different polymerases. So let's keep simple the way it is. All right. Here, this transcription is going to happen inside of the nucleus. However, when this mRNA is ready to be translated, this RNA has to leave the nucleus and go to the cytoplasm. Okay? Here we use RNA polymerase. Remember, polymerase means synthesis, right? It is synthesized RNA. Here, we also have the participation of transcription factors that bind to the DNA, to that promoter sequence, and will control the regulation. It will control the transcription. We have transcription factors that are enhancers, and we have transcription factors that are silencers. As you can imagine, the enhancers, they signal to the RNA polymerase to start the transcription of that gene. And the silencers, they tell the RNA polymerase to not transcribe that gene. Okay? But just remember, transcription factors, they regulate the expression of genes. They are genes that regulate the expression of other genes. That's the main definition of a transcription factor. All right? they have become they have become really important in the last few decades. The more the molecular biology advances, the more we know about these transcription factors, the more we learn about their diversity and their functions. Right? Some of them are involved with silencing genes that um, code for cancerous proteins. Some of them are involved with enhancing genes that uh, generate specific parts of our bodies. So they've been in a hotspot of science over the last few decades. All right. One of the things I do want you to understand is that once the mRNA is transcribed, so the DNA is transcribed into RNA, the RNA is not ready. The RNA has to be processed. So besides of the transcription, there is a RNA processing step here that guarantees that this mRNA is ready to be translated into a protein. What are the processings involved in this step? The first one is the addition of a 5 cap. So this 5-line cap is added to the 5 end of the mRNA. And this 5 cap is a modified guanine nucleotide. And then this cap protects the mRNA from nuclease attack. Those nucleases are enzymes that otherwise would degrade the mRNA. So this cap keeps the integrity of the mRNA. Okay? Another processing that happens is the addition of a poly A tail. The poly A tail is an addition of a 50 to 250 adenine nucleotides to the 3-line end. And this also helps protecting the mRNA from the action of other enzymes that otherwise would attack the mRNA. The mRNA itself, the portion that will actually include and be include co- codons that can be translated into proteins are is very small. But this processing, this addition of other things to that mRNA is important to guarantee that this mRNA is intact and can go to the cytoplasm intact. All right. For the addition of the poly A tail, the mRNA needs a polyadenylation signal, which is the sequence at the three-line end of the mRNA that signals to the cell for the cell to add this poly A tail. Okay, so this polyadenylation signal is important because the cell uses that to recognize the end of the mRNA and knows that it has to add a poly A tail to that end. Okay, the gap, sorry, not the gap, the 5 cap, as the name suggests, is added to the 5 end of that mRNA, which is the opposite side of the 3 line. All right? That's why it is important that you know what is the 5 line and what is the 3 line ends of those molecules. Okay, and I explained that using the DNA as an example. There are three types. There are three types of RNAs. So one of them we've talked is the mRNA. The mRNA is known as the messenger RNA. So the messenger RNA serve as a template for protein synthesis. So the mRNA carries the genetic information used to synthesize the proteins, used to manufacture those proteins. However, we have two other types of mRNAs. We have the ribosomal RNAs and we have the transfer RNAs. The ribosomal RNAs, they are structural components of the ribosomes and they are used for protein synthesis during translation, right? And the tRNAs or the transfer RNAs, as the name suggests, they carry the amino acids to the ribosomes, um, aiding or helping with the protein synthesis. All right. So the mRNA has the information for the protein synthesis. The rRNA or the ribosomal RNA helps with the protein synthesis, and the tRNA or the transfer RNA carries the amino acids, and when they are put together, they make up a protein. All right. So here, when we have a mRNA, this information is written in triplets, which is a group of 3 letters. And that information coded in those 3 letters is called codons. So the codons are groups of 3 letters, okay? And each group of 3 letters, each codon is specific for a amino acid, okay? When you put all those amino acids together, you make up a protein. So proteins are macromolecules. The subunit of the macromolecule is a amino acid. Okay? The amino acids, they follow the sequence of the codons, which are groups of 3 letters of triplets in the mRNA. And here's what I am telling you. Let's look with more details to how the cell makes up the protein. Remember, the process of making protein is called translation. Okay, in this process, the mRNA leaves the nuclei already processed with the 5 cap and the poly A tail. Leaves goes to the cytoplasm. In the cytoplasm, the RNA, the mRNA finds the transfer RNA and the ribosomal RNA. Okay, as you can see here. So here we have the mRNA, we have the transfer RNA, and we have the ribosome, which is this brown big thing here. First of all, the mRNA has the genetic information for making the protein, and the genetic information is in the form of codons. Codons are groups of 3 letters. Okay, those 3 letters are recognized by transfer RNAs. The transfer RNAs, they are in the cytoplasm, and there are many in the cytoplasm here. I'm just not showing them to you. The transfer RNAs, they have 3 letters that complement the codons of the mRNA. So the transfer RNA, they complement the mRNA. And the transfer RNA, they carry the amino acids. Okay? Each codon is specific for one type of amino acids. And then we're gonna look at those codons and those amino acids as well. Here's what's happened. Here's what happens during translation. You have a molecule of mRNA. The molecule is within the ribosome. Okay, as you can see in this detail here. So you have the molecule of RNA, and you have the ribosome, which is attached to the molecule of the mRNA. The ribosome has 2 sites, or sorry, has 3 sites. Has a exit site, has a P site, and has the A site. The A site is the arrival site. The P site is where the polypeptide chain is being formed. And the exit site is where the transfer RNA exits the complex. Okay? The transfer RNA carrying the amino acid enters the arrival site in the ribosome. The arrival site contains, the arrival site contains the 3 letters here that are complementary to the 3 letters to the codon on the transfer RNA. Then what you can see is that this transfer RNA binds to that mRNA. Okay? The transfer RNA that was already there from the preceding reaction is holding the whole peptide chain together. However, that peptide chain is now transferred to the new transfer RNA that arrived at the arrival site. Okay? Now, with a chemical reaction, there is a peptide bond formation here. When the peptide bond forms, look what happens. Now, this old transfer RNA has no peptides, has no amino acids bound to it anymore. Now they have been transferred to the new one that just entered that complex. This guy now goes to the exit site, as you can see here, and leaves the complex. And the transfer RNA now moves to the P site. The P site, as the name suggests, P stands for polypeptide, is the site where the elongation of the chain occurs, okay, of the amino acid chain occurs. This process requires the use of energy. So you need energy to bind one amino acid to the other, and you need energy to transfer or to move those RNAs, those transfer RNAs, across the mRNA. Okay? So energy here is used for the for the binding of the amino acids, which is a peptide bond formation, and it's also used for moving the transfer RNAs from one site to the other. Okay? One key thing that you cannot forget here is that this transfer RNA has a codon sequence that is complementary to this sequence um, on the mRNA. All right? And this transfer RNA is carrying the amino acid. Good? Once you have this chain forming, you have a long sequence of amino acids. And that long sequence of amino acid then assembles into different formats and they form the proteins. We're gonna get there. Okay? Like I said, like I said, the codons, they carry the information or they are specific for certain amino acids. Okay, so you have here this big box showing you that when you have this codon or these codon, remember codons are three letters together, are triplets of letters together. When you have this or that, you have the production of this amino acid. Okay? When you have, for instance, this sequence of codons, you have the production of this amino acid. You have in total 20 amino acids that can be produced depending on the sequence of those codons. How do I read this big box? To your left, it's my left here when I'm looking forward, when I'm facing the computer, when I'm in front of the computer. So when you look at the one axis to the left, this is telling you what is the first codon, the first letter in the codon. Then when you look at the one above, you see the codon that occupies the second position, or the letter that occupies the second position in the codon. And when you look to the right, you look to the letter that occupies the third position in the codon. Okay? So if you have U, 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 you have this codon here. If you have U, U, C, you have this other codon here, and so on and so forth. Okay? You can see that the same amino acid can be codified by different codons. Okay? Remember that information. Another information here important is that you have codons that are specific to initiate the translation and codons that are specific to terminate the translation. When the ribosome finds the codon AUG, then that gives the signal to initiate translation. All proteins have a methionine amino acid. So when the cell or when the ribosomal complex finds this amino acid, then this amino acid, find this codon. This amino acid is inserted in the sequence and translation starts. The same way you have amino acid, you have codons that stop translation. And they stop translation because they do not code for any amino acid. They don't produce any amino acid. All right. And they are UAA, UAG, and UGA. They are known as stop codons because they lead to no amino acid insertion in the sequence. All right. So initiation starts with this, termination ends can end with one of these three types of amino acids of codons. Okay. The same information is shown here. So the methionine is the first or initial amino acid that is present in all proteins because that codon is the first codon that gives the start for translation to begin. Okay, here all proteins are gonna present this amino acid. All right, and it's the only codon that codes for the amino acid methionine. No other triplet of letters code for that amino acid. For termination, we have three codons, and that's because they don't carry any amino acids. Okay, they do not code for any amino acids, meaning that they are not recognized by any transferred RNA. Okay, then, when the cell finds that termination codons, when the cell finds those termination codons, termination or translation terminates. Okay, because there is no amino acid that can be incorporated. All right. There is one problem with these termination codons. When these one of these termination codons are or is inserted in the middle of a gene, then translation terminates, right? If that is inserted in the middle of the gene, that means the protein won't be entirely translated. So only part of the polypeptide chain is synthesized, right? Is terminating earlier than it should have. This type of mutation or this type of change is known as a nonsense mutation. It does not make sense to finish the protein production right here, but the codon is there, so I'm gonna have to finish anyways. So that's why it's known as or it's called as nonsense mutation. Okay, we're gonna come back to this concept, but I wanted to bring this to your attention already. Okay, translation leads to the synthesis of proteins, right? And proteins are important because they make up for lots of biological functions that happen in the human body. You might have heard that enzymes are proteins, right? All the enzymes in the body are proteins. But not all proteins are enzymes. Remember that. You might have heard that in your first biology classes. All enzymes are proteins. Not all proteins are enzymes. Okay? Those enzymes, they are the largest category of proteins. And the enzymes are important because they speed up reactions. They make the reactions happen in a faster manner. But we also have other types of proteins that participate in other functions. For instance, we have the hemoglobin, which is the protein that carries the oxygen in the blood. We also have insulin, collagen, actin, and myosin. Actin and myosin, for those that might take physiology in as part of your degree, as part of your major, actin and myosin are the proteins responsible for making our muscle to contract. Okay, so proteins have this functional and structural role in the human body. Proteins, they are assembly of peptides of amino acids. Okay? Those proteins can exist in different forms, okay, or different structures. A primary structure is um, is accomplished by the peptide bond formation, which is what we just saw. A secondary structure of a protein is when other bonds, such as hydrogen bonds or van der Waals forces, they shape that protein, they shape that sequence in a alpha helix conformation or a beta pleated sheet. Okay? Then you have a third level or a third structure or a tertiary structure, which is when those alpha helix and beta sheets they are assembled together. And when that becomes more complex, when two or more tertiary structures they are grouped together, then you have a quaternary structure, and then you form a more complex protein. Okay, I know might be a foreign concept, so I want to make sure you are familiar. You know what we are talking about. So here, I want to take a moment to kind of review some of those concepts that you might have learned, you know, when you talked about macromolecules, specifically about the proteins, right? So I just said in the previous slide that those proteins can be assembled in different structures. And the first one is the primary structure. And that happens when two amino acids are bound together, right? And we call that bonding as a peptide bond. And that peptide bond causes the release of water in that formation. Okay. So for the first, the primary structure, I want to briefly represent here what's happening with those amino acids. So you have the central carbon of one amino acid, right? And you have the central carbon of the second amino acid. So the R chain that I am representing here is the side chain of that amino acid. But the C is the carbon because remember, we have a central carbon in that amino acid, right? So these amino acids, they have one side with the carboxyl group and the other side with the ammonium group, right? With the side with the ammonium group has something like this, right? And the side with the carboxyl group is something like this, correct? Here, when you have a peptide bond, you have these two guys leaving the molecule, and you have the formation of water. Then, these two amino acid now connect with each other. When you have multiple amino acids connecting to each other, you form a polypeptide chain, right? Because there are many amino acids linked, bond together. And the bond between those two amino acids is called a peptide bond. Okay? Now, this is the primary structure when you have a long peptide chain. The secondary structure now happens because those side, and I'm going to represent with a different color, those side chains might interact with each other. Remember, you have amino acids that are hydrophobic, hydrophilic, polar, apolar. You have different type of amino acids, right? And they might interact with each other. Besides of that, you also have hydrogen in some of these amino acids, in those amino acids, right? And those hydrogens, they like to form what? Hydrogen bonds. So those hydrogen bonds, they help changing the structural conformation of this primary structure, leading to more complexes structures, okay? The next step in that complexity is this secondary structure when you have the formation of the alpha helix and the beta sheets. Then, when you have those interactions, when you have those bonds become stronger, and you have other interactions such as the van der Waals interactions, then you have the formation of tertiary structures. Okay, when you have two or more tertiary structures bond together, then you have a quaternary structure, which is a full, which is a full protein. And here I'm going to represent in a very poor way. So let's imagine that this is one tertiary structure, this is another tertiary structure, this is another tertiary structure, and this is another tertiary structure. So here you have four, so let me use this. You have in total, a total of four subunits together. This is an example of what happens with the hemoglobin. So the hemoglobin is a protein that carries the oxygen, okay, in your blood. The hemoglobin is an assembly of four subunits, and those subunits, they interact with each other due to hydrogen bonds, van der Waals forces that keep those subunits all together. In the hemoglobin protein, we have two alpha subunits and two beta subunits, and they form one entire hemoglobin protein. Okay? So try to picture that as you move from primary to quaternary structure, you are increasing the level of complexity of how those bonds are connecting those molecules together. Okay? This picture is just to illustrate that complexity and how one structure can interact with each other, forming a more complex uh, structure. Okay, so in this example, you have structures right here. This one is different, and this one is another one, forming a entire protein. All right. And why is that important? And why did I bring the case for hemoglobin? Because there is a disease known as the sickle cell anemia. The sickle cell anemia is a mutation or is a muted form of the hemoglobin, and that causes or that impairs the transport of oxygen in the blood. So the person is sick because the cells, the red blood cells, can't carry the oxygen. Okay? This mutation changes the beta hemoglobin. Remember, I told you that hemoglobin is composed of four subunits, but the mutation changes one of the subunits of the hemoglobin. And that happens because one of the amino acids in that um, chain is replaced. So one amino acid being replaced results in a mutant form of that hemoglobin. Okay? That mutant form of hemoglobin can't carry the oxygen, and therefore people are sick. Here's what's happening at the genetic level. And pay attention because this is in one of the um, case studies that you have to turn in. Okay? So here, let's look at it. So here is the sequence of a normal hemoglobin. Okay? A normal beta globin or a normal beta hemoglobin, which is one of the subunits of the full hemoglobin. So the DNA has the following sequence, as you can see here. The DNA is translated to mRNA. Sorry, is transcribed to mRNA. I am very sorry about this. Is transcribed to mRNA, and that's the normal composition of the mRNA sequence. Then the mRNA has the information for for synthesizing a protein, and that is the process called translation. Okay, so this mRNA gives rise to the following sequence of amino acids. Okay, you can see that here the fourth, fifth, sixth, and seventh amino acids are all normal. However, look to what happens with the mutant form of hemoglobin. With the mutant form of hemoglobin, you have the codon here changed. So instead of having CTC, cytosine, thymine, and cytosine, you have adenine. So you have a replacement of only one nitrogenous base. With that, look, you change the mRNA produced, which then changes the amino acid that is coded by this sequence. So instead of having this amino acid, now you have a valine amino acid. However, this is not good, right? Because this is not the original sequence of amino acids of this protein, as you saw here. Because now you have this amino acid, now the protein loses its conformation. And because the protein loses its conformation, it can't execute its function as it did when it was healthy and normal. Okay? For this to happen, you need two copies of the mutant gene um, in the genome of the person. All right. Only one copy does not lead to this um, scenario because this is um, a recessive genetic condition. Okay. Keep this in mind, and as we progress with these classes, you will make sense of these concepts, and you're gonna use this to um, respond to one of your clinical case studies, right? Here, oops, here is a picture of what that mutated hemoglobin cause to a red blood cell. So at the top here, you have a normal red blood cell, which has this round circular shape. However, when the hemoglobin is mutated because replace the amino acid, now you have this hemoglobin, sorry, now you have this red blood cell that can't carry oxygen as efficiently. Okay, so this causes lots of issues for the people that um, have those conditions. All right, now that we have studied um, the processes involved with the genetic material involved with, you know, creating a new DNA strand, creating a mRNA, creating a protein. Now let's look at what happens with the cell cycle and what happens with those chromosomes. Okay, this is a figure of a cell that is showing you the nucleus stained in purple. Here we can see the chromosomes condensed. So each of these worm-like structures that look like little snakes, they are condensed chromosomes. Okay, and everything that's around here is the cytoskeleton. This cell is getting ready to divide. Okay, so here's the nucleus, and outside is the cytoplasm, and the outermost layer is the cell membrane. Okay, a few concepts that I want you guys to understand. So here we have a cell. Inside of this cell, I am showing you a condensed chromosome. But this chromosome is unduplicated or not replicated. Okay? This chromosome has a centromere. Center means central means the center of the chromosome. Okay? Above and below this chromosome, we have the chromosome arm. What happens here from one diagram to the other is the chromosome duplication. So one chromosome now duplicates. Even though it is duplicated, I still have one single chromosome. The amount of DNA changed because now it's duplicated, but the number of chromosome is the same. It is still one. Each half of this chromosome we call sister chromatids. Okay, so here we have two sister chromatids in the same cell. Then the next diagram is showing you these sister chromatids separated. Okay, so this is what we are going to study, how this chromosome from duplicated give rise to other cells, studying the cell cycle. Before we go to to those details, we know that the chromosomes receive names according to the position of the centromere. Remember, the centromere is the middle, that constricted region in the chromosome. This centromere can be located in the middle or between the middle and the end, above or below the center, can be close to the end of the chromosome or at the end of the chromosome. Depending on which position the centromere occupies, the chromosome is then called the chromosome is then called metacentric, submetacentric, acrocentric, or telocentric. Let's look at the picture of what they look like. So here is a metacentric chromosome, meaning that the centromere is in the middle. This is a submetacentric chromosome, meaning that the centromere is now between the middle and the end, right? Is off the center. That's why sub is off the center. Then the third example is the acrocentric, when the acrocentric mean is close to the end, means that the centromere is close to the end. And then telocentric is when the centromere is all the way to the end. Telo means end in biology, or at least in cellular biology. Okay? These chromosomes, they exist in pairs in most of the diploid cells of the human body. A diploid cell means that the cell has twice the number of a haploid cell. Haploid cell has half of the number of chromosomes. In humans, a diploid cell has 46 chromosomes, and those 46 chromosomes is identified as 2n. So you can simply say that 2n equals to 46. If 2n equals to 46, then a haploid, which is half, should be 23 chromosomes. Okay? These chromosomes, they exist in pairs. So remember, and I'm going to show you a figure of this karyotype again. Remember that in each of those pairs, I showed you two chromosomes, right? They are called homologous. They are called homologous because they are similar. They carry similar genetic information. Let's look at the karyotype again. So this is a karyotype. What you can see here is the 23 pairs of chromosomes. So this is the third pair of chromosomes. You can see that I have one pair and sorry, that I have one chromosome and I have the second chromosome. These two chromosomes are called homologous. Okay? Because one came from the mom and one came from the dad. One comes from the biological male and the other comes from the biological female. Each chromosome, and now I'm going to show you in different colors, has sister chromatids. Okay? So this chromosome has two sister chromatids. Oops. So that chromosome has two sister chromatids. And this other chromosome has two sister chromatids, okay? But they are homologous to each other, which means that one came from the mom and the other came from the dad. So they carry similar, not the same, similar genetic information. What do I mean by that? Let's do this. So, if I have one chromosome that came from my father, the other chromosome came from my mother, okay? These chromosomes are duplicated. How do I know that? Because they have sister chromatids. All right. Let's say that my mom has the information for blue eyes, and I'm going to represent that with capital or with lowercase a. Don't mind about this detail. We're going to talk about this in the second class. And my father has the information for brown eyes. Okay? You can see that I purposely wrote these A's at the same level in both chromosomes. Why? Because they are homologous, meaning that the information about the eye color is in the same position of those chromosomes. One came from the father, the other one came from the mother. But the information about the eye color occupies the same localization, the same position in both chromosomes. This position is called locus. Okay, locus is the position that the gene occupies in the chromosome. The plural of that is loci. Okay, but here is the thing. I told you that they are similar but not identical because see that the genes are different. I told you that the father is carrying color for brown eyes, the mother is carrying color for blue eyes. They are both eye colors, but they are coming in different flavors. Does that make sense? So those colors are not the same, they are different. Okay, but they are both eye colors. Those are two homologous chromosomes because they carry similar information, but the genes can come in different flavor. And those flavors, I know this is very dumbed down, but those flavors are called alleles. Okay, so here my father has this allele, and my mother has the other allele. Okay, the allele is the flavor of that gene that can be different. Locus is the position of that gene that is the same if they are homologous. Okay, these are two duplicated chromosomes because they have sister chromatids. I want you to repeat that information until it makes sense in your own head. What is sister chromatids, homologous chromosomes, locus, alleles, genes? Those concepts, they have to be um, part of your daily studies for genetics because they will come up in later classes. And it is important that you understand this now because when I say, oh yeah, this trait occupies the locus, whatever, in the chromosome, you know what locus means. Locus means place in the chromosome, localization. Think about this. Not localization, sorry, location. Or you understand the how this would go. Okay? Haploid cells is when they have half of the number of the diploid cells. Okay? For humans, as you can see here, the haploid number for humans is 23 because the diploid number is 46. Okay? Where do we find haploid cells? Those haploid cells in humans are the gametes. Okay? In plants, they are the spores. Okay? But they form the gametes of the living beings. All right? Oops. This slide is only going through those terms I told you already. So, like I said, locus refers to the place of the gene site in that homologous, in those homologous chromosomes. Okay? Allele is the flavor, is the other format of that gene. One gene can code for brown eye, the other gene can code for blue eye. Okay? And that's what we call biparental inheritance. Biparental means we have two parents. Okay? So one information comes from one parent, the other information comes from two parent from the other parent. The combination of these two information gives the phenotype. Okay? The genotype, and we are going to come back to this concept in the second class, is the genetic information within those homologous chromosomes. However, is the phenotype is how the genetic information is expressed with the environmental clues. Okay, we're gonna go back to these two concepts, but this is just going over what I explained to you in my previous diagrams. Humans also have what we call a non-homologous, oops, non-homologous chromosomes. And those are the sex-determining chromosomes. They are non-homologous specifically in males. Okay? In females, those chromosomes are XX. So they are homologous, they are similar. But in males, they are XY. Usually, the X chromosome is smaller. It's usually smaller, okay? And it doesn't contain most of the genes that are in the X chromosome. That's why they are not homologous because the X is different from Y, okay? Now that we are familiar with these process that we've been describing, um, over the past few slides, we can talk about the cell cycle. All right. That's, you know, how the chromosomes are going to be divided in the cells. Okay. The cell cycle is composed of two main phases. All right. We have the interphase and the mitosis or the meiosis. Right? So one cell cycle has interphase, mitosis, or meiosis. We're going to start with mitosis. And most of the processes that I explain here for mitosis also works for meiosis. The difference, like I said, is the outcome. It's going to be the number of chromosomes at the end in the daughter cells. All right. So, cell cycle is composed of a interphase, and here starting with the mitosis. The interphase can be further divided in G1, S, and G2. The G phases here stand for gap. That's when the cell kind of stops and checks if everything is okay to proceed for the next step. Okay, so let's understand these gap points as checking points for the cell before proceeding to the next phase of the cell cycle. The S phase here is the phase of DNA synthesis. That's when the amount of DNA in the cell increases, or that's when the DNA duplicates. It's during the S phase of the interphase. Okay, the interphase is the longest. Is the phase that takes the longest time to be completed. Mitosis and meiosis usually happens in matter of few minutes to hours. Okay, the interphase can take much longer. All right, and I'm gonna show you in the next few slides um, how long it might take. All right, so here you can see that the G1 S and the G2 phase, they take up most of the cell cycle. And the mitosis is actually very short, right? The G1 phase and the G2 phase are gap phases where the cell checks if everything is okay and if the cell cycle can continue. The S phase is when the cell is actively synthesized, duplicating the chromosome. Okay? Then the mitosis is when the cell actually divides. And the phases of mitosis are prophase, metaphase, anaphase, and telophase. I know I am showing you a pro-metaphase here, and I am going to come back to this later on. But here, what I want you to know is have this dimension, is have a visual sense of how the interphase occupies most of the cell cycle of the cell. Of the cell cycle of the cell seems redundant, but you know what I'm saying. In terms of hours, here's an example, okay? So mitosis, if we were to break down those phases, they only take a few minutes each phase. However, the interphase, each of the phases in the interphase can take more than hours to be completed, with the S phase being one of the longest phases of the cell cycle. All right. The first phase, as you can see here, is the interphase. When the chromosomes are not condensed, they are extended and uncoiled, and they are found in the form of chromatin. Here they are not duplicate. Here they are duplicated already, but they are not condensed. Okay. Then we have the mitosis, which is divided in the following phases. And we are going to look at those phases one by one. This diagram is showing you all the phases together. So after the interphase, the cell goes to prophase. Then the cell goes to prometaphase, which is a preparation to metaphase. Then goes to anaphase and then telophase. You can see at the bottom a diagram, a illustration of what we can actually observe under the microscope. So these figures here are actual microscopy figures of what we can observe, of the cells that we can observe um, under microscope. Okay, how the cells look like. Let's look at each phase individually. The first phase is the prophase. In the prophase, you have the chromosome condensed. Okay, so you can actually see the chromosomes. This is a fluorescent preparation, so you can actually see those chromosomes a little bit better. But in our diagram, you can see the chromosomes already condensed. What also happens here is that the centrioles, they move to opposite sides of the cell, to opposite poles of the cells. Okay, and um, those poles are called the centrosomes. Those centrosomes, they are in two different poles of, they occupy two different poles of the cell. Okay, during the prophase, besides of the chromosome condensing, also the nuclear envelope starts to break down, because these chromosomes will have to be reached by the cytoskeleton so they can be pulled apart. Okay, besides, during the prophase, we also have the formation of the chromatin fibers that now are visible chromosomes. Okay, because they became condensed. We also have the formation of the mitotic spindle. So these are microtubules. This is the cytoskeleton of the cell that would be responsible to pull the um, chromosomes apart, all right, during the metaphase. So prophase, first phase of the mitosis, right? During that first phase, the chromatin now is become more condensed and the chromosomes are now visible. We also have the movement of the centrioles to opposite sides of the cells, occupying the centrosomes, which are different poles of the cells. Okay, we also have the breakdown of the nuclear envelope, because at some point these chromosomes, they will have to be reached and then pulled apart. Okay, the second phase is the metaphase, that sometimes we can break down into prometaphase and actual metaphase. In the prometaphase, those chromosomes now, they start organizing, they start moving to the center of the cell. How does that occur? Well, the cell has um, cytoskeleton, and the cytoskeleton moves those chromosomes, rearranges those chromosomes so that in the metaphase they occupy the metaphase plate, which cuts the cell in half, essentially. The metaphase plate is just a illustration of the chromosomes occupying the center, the middle of the cell. Okay? During the prometaphase, now the nuclear envelope is completely or almost gone. During the metaphase, the nuclear envelope is completely gone. Here, you have the centrioles or the microtubules already migrating to the kinetochore. So you have the extension of these spindle fibers binding to the center of the chromosomes, of the centromere of those chromosomes. And that is important because that helps moving the chromosomes. Okay? In the metaphase now, the centrosomes are already in opposite sides of the cells. And here you have the microtubules reaching the chromosomes. Okay, and those chromosomes are aligned in the middle of the cell. I mentioned a word, or I mentioned the existence of the kinetochore, right? The kinetochore is where the spindle fibers connect to the chromosome. The kinetochore allows for the junction of those spindle fibers to the chromosomes. And that is important because these chromosomes are going to be pulled apart during the anaphase, which is the next phase of the cell cycle. What is important here to observe is the presence of a few proteins, and they are the cohesin and the sugosine. Those proteins, they are responsible for keeping the sister chromatids together. As you can see, the cohesin is keeping the sister chromatids together through the arms, and the sugosine is keeping those cohesins together through the kinetochore. Okay? However, when these sister chromatids start separating in the anaphase, you have an enzyme that degrades the cohesin, and that enzyme is called the separase. As the name suggests, it separates. Okay, separates the sister chromatids, which is important for the next phase of the cell cycle, which is the anaphase. Here, during the anaphase, we have the separation of the sister chromatids. So here, you can see that half goes to one side of the cell and the other half goes to the opposite side of the cell. Okay? So they are no longer at this point sister chromatids, but now they are called the daughter chromosomes. Okay? This is only accomplished because those enzymes, such as the cohesin, is degrade or cleaved by the separase. Remember, the cohesin is keeping the sister chromatids together. Okay? Also, another enzyme that degrades is the sugosine. The sugosine is keeping the kinetochore connected to the cohesin, keeping the centromere connected. All right. But during the anaphase, all that degrades, the centromere splits, and those sister chromatids can be separated. Okay. Ana means separation. And this is a very short stage of the mitosis. All right. Here, what is important to notice as well is that those microtubules are pulling those sister chromatids or the daughter chromosomes away from the metaphase plate to the opposite pole of the cell. Okay? The last phase that we have here is the telophase. During the telophase, what happens is the division of the two new cells. And that division we call the cytokinesis. So cyto means cells, kinesis means division. So here we are dividing the cytoplasm into two different cells. What also happens during the telophase is that the chromosomes, they now uncoil and they form the chromatin again. Remember, the chromatin is just the DNA not condensed. Okay, so the DNA is just spread through the nucleus without being in the format of chromosomes. Here you can also see that during the telophase, the nuclear envelope reappears and the spindle fibers or the microtubules that pulled the chromosomes apart, they disappear, okay? And the cell now divides, all right? So chromosomes are less condensed, cytoplasm of both cells are dividing, the nuclear envelope is reappearing, is being formed again, and the chromosomes are now chromatin. Okay? Cytokinesis, as you can see at the bottom here, is the complete division of the cell. Okay? It's usually followed by the telophase. Hard to differentiate them. But as long as you imagine the cytokinesis as the separation of the cells and the telophase as the whole thing, I'm happy with. Remember that I told you that the cell has checkpoints during those gap periods. Here, this picture is showing you that the cell has three major checkpoints. One of them happens in the middle of G1. The other one happens at the end of G2. So they happen at the gap phase of the cell cycle. And that is important because the cell wants to make sure that everything is okay before it proceeds to the next phase. So here, this checks the cell before the cell goes to the S phase. And here is checking the cell before the cell actually goes to mitosis or meiosis. Okay? If the cell cannot proceed to the following phase, then it goes to a period that we call G0, where the cell is hopefully killed. All right. If the cell is not killed, then it can become a tumor. Right. So we expect the cell to be arrested at those checking points to prevent bad things from happening. All right. So besides of those checking points that I showed you in the previous slide, I also want to show you that the cell uses other signals that tells the machinery in the cell of to start those cell cycles, those divisions. Okay, one of them is the production or the synthesis of this. Um, where's my laser? Of this protein called cyclin. The cyclin starts being produced at the end of the S phase. So the cyclin starts being produced at the end of the S phase and is produced all the way through the G phase. At the same time, you have the production of the CDK. The CDK is a cyclin-dependent kinase. The cyclin-dependent kinase, as the name suggests, is dependent on the presence of the cyclin. When the CDK and the cyclin are present, they combine and they form the MPF complex. The MPF complex means the maturation promoting factor. The maturation promoting factor, as the name suggests, promotes mitosis. because this factor or this complex will induce the phosphorylation of different proteins involved with the mitosis. So by the end of the G2 phase, the levels of MPF are high, which stimulate the initiation of the mitosis. So this is the molecular mechanism that starts um, or that signals for the cell to proceed with the mitosis. Okay, the MPF levels are the highest during the metaphase, and then after the anaphase or during the anaphase and right after the anaphase, the complex, especially the cycling, starts being degraded. And without this, the CDK can't function and it is recycled for another cell cycle. Without this complex, then the cell stops dividing and enters in the G1 phase, in the gap phase. Okay, so elevations of cyclin and CDK leads to the formation of the MPF, and the MPF promotes mitosis due to the phosphorylation of proteins involved in the mitosis phase. Okay? However, at the end of mitosis, around anaphase telophase, the cyclin is degraded, meaning that the levels of cyclin diminishes, the CDK is not functional, and now the cell finishes the cell division and then enters in a G1 period again. Okay? The next step I want you to, or the next thing that we should cover is the meiosis. The meiosis will create, like we said, haploid cells. And those haploid cells are the gametes. And that is important. Meiosis is an important process that guarantees generic variation in the species. Okay? You will see that I am going to skip those processes, prophase, metaphase, anaphase, and telophase, because they essentially happen in the same way that they happened in the mitosis. What I want you to understand are few key things that happen in the meiosis, and that's what I'm gonna stress out here in this um, coming slides. Okay, so moving on. Here, let's remember some key concepts. The blue represent those chromosomes that came from the father, whereas the red represents those chromosomes that came from the mother. Here you can see that the diploid number is equal to 6, meaning that there are 3 chromosomes that came from the father and 3 chromosomes that came from the mother. Okay? This chromosome is paired to this chromosome is paired to this chromosome, and this chromosome is paired to this other one, as you can see. If the diploid is 6, the haploid number is 3. Okay? Because the haploid number is half of the diploid number. Okay? What I also want you to understand is that we have 3 pairs of chromosomes, but we have 6 chromosomes in total. Okay? So this is one pair, this is the second pair, this is the third pair. If they are pairs, then we say that they are homologous. So these two is one pair of homologous chromosomes. And you can see that each chromosome, they are duplicated. So this and this are sister chromatids of the same chromosome. I know that this might sound repetitive, but it's important for you to make sure that you understand those concepts because, you know, many years teaching these to my students, they do struggle to understand what is a sister chromatid, what is a homologous chromosome, what is a pair, what is a haploid number, what is a diploid number. So I do want to make sure that you understand those concepts by repeating over and over, giving you different examples of what that um, looks like. Okay, so the meiosis is a type of cell division that produces the gametes in the animals or the spores in the plants. The spores also form the gametes then in the plants. Okay, they only form haploid number of chromosomes, meaning that the number of chromosomes is cut in half. Because of that, we say that the meiosis reduce the amount of the generic material in the cell. The material comes back to 46 or to whatever the diploid is when fertilization occurs. Okay? The meiosis The meiosis is important because it maintains the generic variability. Okay? And the generic variability is accomplished through the crossing over. The crossing over happens between the homologous chromosomes and they exchange generic material with each other. Okay? This crossing over allows for generic variation because those chromosomes are exchanging information with each other. All right? And that's what I want you to know. Where does that crossing over take place in the meiosis phases? Okay? And we're going to look at that shortly after a few slides. Okay, there are two meioses. Okay, the first meiosis or meiosis 1 is known as the reduction division. The second meiosis or meiosis 2 is known as the equational division. Both meiosis 1 and 2 have the same prophase, metaphase, anaphase, and telophase stages. Okay, so instead of going through each one of these phases, I want to give you a general picture of what happens to the number of chromosomes in each phases. I do want to call your attention to the prophase of the meiosis because it is during the prophase 1 of the meiosis 1 that the crossing over occurs. So here you can see two homologous chromosomes. And here you can see that they are crossing over their arms. It's literally putting the arm over the other. They are crossing their genetic information. At the end, see that the orange chromosome now has a piece of the blue chromosome, and the blue chromosome has a piece of the orange chromosome. So genes that were not carried by the orange chromosome now are being carried by the chromosome, and vice versa. So that is why the crossing over during the prophase 1 of meiosis 1 is important for generic variation. because it allows for exchanging genetic information. And the genetic information that chromosome did not have, now it does. Okay? That region, that crossing over, we also call chiasma. Chiasma is the formation of this X, this crossing over between those two homologous chromosomes. Now, I want to trick your head, and I'm sure that you guys are very smart, and this is not going to trick you in any means. But, question to think: can this crossing over happen between two sister chromatids? And if it does, is there any value of this happening? Think about it. If we're talking about generic variation, would that actually be relevant for the cell? Crossing over with the same sister chromatid? Maybe not. Maybe yes. I don't know. Think about it. So, the meiosis one is a process where we have the separation of the homologous chromosomes. Okay? We have all the events, all the stages happening. We have the prophase, metaphase, anaphase, telophase. But they receive the number one because they refer to the first meiosis. Okay? But at the end of meiosis one, and this is what I want you to remember, is that you have the separation of the homologous chromosomes. How is that accomplished? Remember, here we have the, well, I guess I am not showing you here, but we have the prophase, right? So during the prophase, the chromosomes become condensed. Okay? Then the next phase is the metaphase. In the metaphase, those chromosomes, they occupy the metaphase plate. So they are in the middle of the cell, right? Those chromosomes are organized in the following manner. You have the same homologous chromosomes paired with each other, but in different sides of the metaphase plate. Okay? So the homologous chromosomes in the metaphase one, they have to be facing each other. Question: can I have this chromosome, this orange chromosome, on the same side of the blue big chromosome? The answer is no, because here I want to separate the homologous chromosomes. If them two are on the same side of the metaphase plate, when the cell separates, they go to the same cell. And that's not what we want to accomplish with meiosis 1, right? Because meiosis 1 has to separate those homologous chromosomes. Okay? So the homologous chromosomes, they cannot occupy the same side in the metaphase plate. They do face each other, but they are on opposite sides of the metaphase plate. Okay? That's the most important thing to happen. Then you have the anaphase, which is when they are pulled apart, and the telophase, which is when the two cells are formed. See what happened now? This chromosome is homologous to this, but they are not on the same, they are not in the same cell. They are in different daughter cells. So these cells we call daughter cells. Okay? And then here At the end of meiosis one, we have the separation of homologous chromosomes. I'm gonna just do like this because I've ran out of space. Okay, so separation of the homologous chromosomes. One of the things that I do want you to understand about this process, about this meiosis, about what happens in meiosis one, is the independent assortment. Like I told you, the big orange chromosome, I'm sorry about that. Like I told you, the big orange chromosome cannot be on the same side of the big blue chromosome because they are homologous chromosomes and we want to separate them. Okay, the cell can't carry two alleles for eye color, right? You're either brown in most cases. Your eyes are going to be either brown or blue. You don't have them together, brown and blue, in most of the cases. Okay, I'm just trying to simplify the process. Here, what can happen is what we call the independent assortment, meaning that the position of these two does not interfere with the position of these other two. So you can have the blue on this side, the big blue on this side, and the big orange on this side, but you can have the little blue on that side and the little orange on this side. Not necessarily you have to have them in this configuration, okay? Because the big chromosome is independent of the small chromosomes, meaning that if they are not homologous, it doesn't matter which side of the cell they occupy. Okay? But if they are homologous, they must be in different sides of the metaphase plate. Okay? So, at the end of meiosis one, we have the separation of the homologous chromosomes. Okay? As I told you, it is in the meiosis one that happens the crossing over. And the crossing over is important because it provides a source of generic variability in the formation of the gametes. Okay? Besides of that, the process of independent assortment and the segregation of the chromosomes also allows for further generic variability of these gametes. And I'm going to talk about these two processes later on in my final slides. Okay? Here is a schematic diagram of the phases of meiosis 1. I'm not going to go over each of these phases because, like I told you, the phases they resemble what happens in mitosis. Okay, what I do want you to understand is that at the end of meiosis 1, we have the separation of the homologous chromosomes. Okay, of the homologous chromosomes. So the outcome here is that at the end of meiosis 1, each cell finishes with 23 chromosomes, 23 duplicated chromosomes. Okay? Because the next step, which is the meiosis 2, now we're gonna separate the sister chromatids. Okay? So meiosis 1 separates the homologous chromosomes. And at the end, the cell, if the cell started and I'm gonna write this down. If the cell started with 46, at the end, each one of them has 23, okay? And each one of them have the chromosomes duplicated because the sister chromatids are still connected, okay? It is in meiosis 2 that we separate those two sisters chromatids, okay? One of the things I was trying to explain you in the previous slide was the independent assortment. What I told you was, let me get the pointer. Here I am representing the metaphase plate, right? And that's when the homologous chromosomes, they align in the middle of the cell. What I told you is that the homologous chromosomes, they have to be separated in different sides of the cell. They cannot be on the same side of the cell. I cannot have the big orange chromosome on the same side of the big blue chromosome. Okay? What can happen is, look at these different cases. What can happen is the order of these two to shuffle. So instead of having blue and orange like this, I can have orange and blue. The same thing can happen with the smaller chromosomes. Instead of having blue and orange, I can have orange and blue. Thinking that these cells are gametes, see that here I generated how many different gametes? I generated 8 different gametes. Okay, so this gamete is definitely different from this gamete. This gamete is definitely different from this gamete. Okay, because of the independent assortment. The independent assortment tells me that the big chromosomes, and I am dumbing this down, the big chromosomes do not depend on the smaller chromosomes. Okay? Their positions are independent of each other. You do want to keep the homologous in different size of the um, in different sides of the metaphase plate. But if the other pairs are going to be on the same side or not, that doesn't matter. Okay? So the independent assortment is important for generic variability because at the end, you can generate 4 different cases of how these chromosomes can be rearranged. Okay, at the end of the meiosis. All right. After meiosis one, we have the meiosis two. Here, remember, we started with one cell, and that one cell gave orange created 2 other cells. Each cell is already a haploid cell with a haploid number of chromosomes. Now, in the meiosis 2, what we want to do is separate the sister chromatids. Remember, this is one this is one sister chromatid, and this is the other sister chromatid. And we want to separate them. Okay? So at the end of meiosis 2, we have the separation of the sister chromatids. Here we have 2 cells, and each cell give rise to 2 other cells. So at the end, we have a total of 4 cells being formed. What about the number of chromosomes? They were haploid here. They continue to be haploid here. only the mother cell was diploid. But then after meiosis one, they were all haploid already. Okay? So at the end of meiosis one, we have the separation of the homologous chromosomes. And then at the end of meiosis two, we have the separation of the sister chromatids. Meiosis 2 also has the same phases as the um, meiosis 1 and as mitosis. It also has a prophase, metaphase, anaphase, and telophase. The difference is that they receive different numbers. Now they are receiving the number 2 in front of their names. During the metaphase, those chromosomes, they line up on the metaphase plane. We don't have homologous here anymore. Then, during the anaphase, the sister chromatids are pulled apart. Then, during the telophase, the cells divide, and at the end, after the end of the cytokinesis, you have those sister chromatids separated in four different cells. Okay? Like I told you, this is just a big diagram that summarizes the prophase, metaphase, anaphase, and telophase of the meiosis 2. At the end, what we are interested to know here is that the meiosis 2 leads to the separation of the sister chromatids. Okay? Again, our cells in the meiosis 2 started with 23 chromosomes and then finished with 23 chromosomes, meaning that we had a haploid cell finishing with a haploid cell. It is in the meiosis 1 that we had a diploid cell finishing with a haploid cell. Okay? That continues to meiosis 2. Again, review those phases with yourself to make sure you are familiar with the processes that happen in each of these phases. Okay? There are two types of gametes that biological females and biological males produce. Biological females, they produce the eggs. Biological males, they produce the sperm. The production of the sperm is known as the spermatogenesis, and it happens in the testes of the biological males. And the production of the eggs or the oocytes happens in the ovaries of the biological females. And the name for that process is oogenesis, right? Here is a diagram that compares, that shows the spermatogenesis and the oogenesis. On your, I believe, left, you have the spermatogenesis process, and on your right, you have the oogenesis process. Let's look at the spermatogenesis process first. Everything starts with a spermatogonium. The spermatogonium is the stem cell, meaning that this cell only undergoes through mitosis. Because this cell undergoes through mitosis, this cell is producing two other cells. One of the cells undergoes mitosis because you need new cells every time, and the other one is selected to go through spermatogenesis. The other one that is selected to go through spermatogenesis is called the primary spermatocyte. The primary spermatocyte undergoes through meiosis 1 and forms two secondary spermatocytes. Again, spermatogonium suffers mitosis. One cell remains as a spermatogonium, as the stem cell that keeps originating new cells, and the other one is selected to be the primary spermatocyte. The primary spermatocyte undergoes through meiosis, one, and produces two secondary spermatocytes. Those secondary spermatocytes, they undergo meiosis, two, and then they produce four new spermatids. So from primary spermatocyte, we formed secondary spermatocyte, and then we formed spermatids. Those spermatids, they have what? N number of chromosomes. Here we had 2n. Here we also had 2n. But after the first meiosis, we have n, and here we also have n number. Okay? The spermatids, they undergo through differentiation and they form the final sperm. The same thing, very similar, happens with biological females. With that process we call oogenesis. Here we have a oogonium, which is the stem cell that undergoes mitosis, forming two new cells. One cell remains an oogonium, and the other cell is selected to be the primary oocyte. The oogonium is 2N, so it's a diploid cell, and the primary oocyte is also a diploid cell, right? Then the primary oocyte suffers or goes through meiosis 1, and it forms the secondary oocyte. Here we have two cells after the meiosis 1. One of the cells is the secondary oocyte, like I said. However, the other cell does not develop and forms the first polar body. Okay? Because this cell does not develop, this cell does not go through meiosis 2. Only the secondary oocyte goes through meiosis 2. And then from the secondary oocyte, the cell goes through meiosis 2 and forms the tertiary, I'm sorry about the name here, the tertiary oocyte. Okay, you can correct the name here. It's a tertiary oocyte. Sorry. Here, remember, one cell produces two. One cell would have produced two. Because one of the cells developed less than the other, this other cell is known as the second polar body. Those polar bodies, they form because they receive less cytoplasm of the cell. They receive the same amount of DNA, but they receive less cytoplasm. And because of that, they do not develop. Okay? At the end, this tertiary oocyte also goes through differentiation processes and form the final egg or the ovum, which I also like to call oocyte. Okay? Um, this ovum will then be combined with one sperm, and then the number of the new cell becomes diploid, right? So let's follow the numbers. First, we had 46. Here, we also have 46. Well, let me do 2n, 2n. Here we only have n, and, and. Same thing to the spermatogenesis. We had 2n, 2n, and, and N. Okay, it is important that you understand what's happening with the number of chromosomes because that helps you understand, that gives you a general idea of the processes of each cell division. The last slide, because I know that you guys are very tired now. I just wanted to mention that, meiosis is important because it is important because it is a critical process for the successful sexual reproduction of animals, of diploid organisms, plants, um, and that allows for generic variability of these um, organisms. Okay, here you can see the whole cycle together on your right. You can see that the ovary of biological females and the testes of biological males contain those gametes. Those gametes, which are the egg and the sperm, are haploid cells, meaning that they only have n number of chromosomes, which for humans are 23. But you can look up for other animals, and I know, and for other organisms, I know I have a table back here in these slides. The n number for different species is different, okay? When these two cells that are N, they combine through fertilization, now we have a diploid zygote. We have a diploid cell, meaning that we have two N, right? N plus N equals to 2N. If each one of them had 23, now we have 46. The zygote goes through uh, different mitosis to various mitosis, and they form a full organism. Okay? What I want you to see is that we developed from a theory that said, oh, non-living things give rise to living beings. For me to show you now that that's not the case, we do need cells, we do need pre-existing organisms that undergo through those successive genetic processes and they combine to form a new organism. Okay? That is true for all of those organisms that are diploid organisms and that goes through sexual reproduction. Okay? This is important as well because it allows for generic variation of the species. And the generic variation would be very important when we talk about the population, the genetics in the population, and how the frequency of the alleles and frequency of those genotypes can be influenced. Okay, so I understand this was a lot of information. I hope you take some time to review this class and go over the main points that you perhaps um, don't understand, and ask me if you have any questions. I'll see you soon.